All right. Time to get started. Before we start, some of you have already heard this, and if not, this is my last semester doing SI for biochemistry, TAing for biochemistry, blah, blah, blah. Actually, technically, the summer semester is. So, if you know anyone that's willing to do SI for biochemistry, that is something I'm actively recruiting people to. I know there are a few people already who have signed up to try and all that, but we need as many as we can get. As you can see, biochemistry is a very difficult topic, and usually most semesters there's one SI and a couple tutors. So if you know anyone that wants to tutor or SI biochemistry, please let me know. Or if you yourself want to, please let me know. Other than that, yeah, this is it. I am definitely not coming back to do this anymore. Please make sure you tell everybody this. And yeah, so it's my second retirement from this thing. What else? Some other announcements. Oh, I will not be here on the day of the final exam, so I won't be there to answer questions like I usually do in terms of proctoring. I'll be away at a conference, so I'll do my best to leave behind people that can try to answer any questions that you guys have during the exam. Carl should be there. If you know Carl, he's the previous TA for the course. He'll be there. Let's see. So I won't be there on the day of the actual final exam. I'll still have the exam review during Up All Night. I'll send that info out in a little bit. Let's see. Other announcements. That's about it I can think of. Did Dr. Easy start going over chapter 30 ahead of time yet, or no? Okay, good. Yes, question. Transcription? It all depends on how quick he gets it done. I will let you guys know during the exam review if it's going to be on there or not. It doesn't hurt to go over it anyway, especially because it'll be on the final if it's not going to be on exam 3. So that's why I'm going to continue with eukaryotic transcription. When we reach the end of 29B, though, I will pause and I'll do some review. And you guys got the email, I'm assuming, for the exam 3 review. That's going to be on Wednesday evening, 6:30 to 8:30. It'll be recorded like last time. What else? Always forget something. After I may start chapter 30 early, so we can get it out of the way. Like, I don't know what SI session yet, but eventually I'm going to be covering chapter 30 quite ahead of time because we need to start reviewing for the final. So on that day that we're covering chapter 30, it's not going to be for exam 3. It's going to be for the final exam. So I'll be doing it a bit ahead of time. How's the studying going? I probably should have been asking this over the last couple of weeks. Is there anything that you guys are finding more difficult than not right now? Who's having trouble with DNA replication? Like a real significant issue there? No, you guys got it. Cool. 100% on that chapter. How about yes? Do you know what the credit is going to be? I don't know the one choice. I just know historically it has been lac operon, it has been describe prokaryotic transcription, the steps, the enzymes, all that, and siRNA, describe the mechanism of siRNA and how that works, how RNA interference works. Those are the 3 historical extra credit questions I've seen for exam 3. So again, for those listening, I have seen the exam 3 extra credit question be the lac operon, be describe prokaryotic transcription, so all the steps and enzymes and subunits, and siRNA, how it's made, what it does. Those are the 3 extra credit questions I've seen in the past. All right. All right, so let's finish up 29B today. You will find that we'll probably be skipping a lot because a lot of it is not necessary for this class. But to pick up where we left off, eukaryotic transcription is regulated in a way more complicated way than prokaryotic transcription was. Prokaryotic transcription, you had the operon, you had inducers and repressors, co-inducers and co-repressors, and you had those four ways of four combinations, right? Positive inducible, negative inducible, blah, 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 blah. In eukaryotic transcription, it's way more complicated because there's way more regulators. And what I mean by that is every single thing here that you see with the word TF is transcription factor. There are, in eukaryotes, there's a category of proteins called transcription factors whose job is to regulate the transcription of genes in eukaryotes. They do it in a variety of ways. Some transcription factors recruit RNA polymerase to the transcription start site. Other transcription factors repress transcription. They prevent RNA polymerase from going to the transcription start site. Other transcription factors adjust the chromatin around a transcription start site. Remember, in eukaryotes, we have histone proteins, right? Anyone remember what a histone is? A histone is a protein that DNA can be wrapped around. And when you have DNA wrapped around a histone, what term do we use to describe that? Nucleosome, right? So there's a category of transcription factors that methylate and acetylate histone proteins to affect transcriptional regulation. So there's a whole variety of categories. Most of, really any of it, you don't need to know for this class. Just understand that there's a huge variety there. The way transcription factors regulate transcription, or where they know how, where to regulate transcription, is via binding to consensus sequences. In eukaryotes, we use the phrase elements to describe these consensus sequences. So let me jump to that slide real quick so you know what I'm talking about. Whenever you see the phrase DNA elements, it is a particular pattern in your genome that transcription factors like to bind to. And when they bind to that particular spot, they regulate the region around it. There's a whole set and there's a whole variety of DNA elements in eukaryotes. You might see these phrases such as promoters, enhancers, response elements, silencers, and initiators. You do need to know the definitions of each of these. You won't be asked to identify one from the other, but just the definitions. When you go look at this picture here, up here at the top, you see some of those DNA elements, or the regions that they would be found in. The site that is proximal to the transcription start site, the region that is proximal to the transcription start site is called the core promoter. The region that's a little bit more distal is called the regulatory promoter. So these are regions approximately a couple thousand base pairs long that within them contain consensus sequences for various transcription factors. The one you should know by name, consensus sequence-wise, is the Tata box. The Tata box is a particular pattern of bases in the core promoter of eukaryotes. Eukaryotes, must stress that. People very often get that confused with the prokaryotic version. As a test, what's the prokaryotic version of a Tata box called? Pribnow box, right? You go review your prokaryote transcription notes, you will find that they talk about the Pribnow box instead. Anyways, in eukaryotes, you have a Tata box. Transcription factor 2D is one of your core transcription factors. It binds to the Tata box, and not by itself, a bunch of other transcription factors have to come in, but collectively they will recruit RNA polymerase to the transcription start site. And when all that mess comes together with transcription factors, co-activators, and RNA polymerase 2, you have what is called a basal transcription apparatus. This machine is going to go and transcribe the eukaryotic gene. Other than core promoter and regulatory promoter, there's another region of a DNA element called an enhancer. This, by definition, what an enhancer is a really far away DNA element. It can be millions of base pairs away from the transcription start site, yet it still interacts with the transcription start site because of this unique feature of eukaryotic DNA, in that eukaryotic DNA is able to bend and loop around itself like so. So although if you count the number of bases it takes to get to the enhancer region, it may be millions of base pairs away, three-dimensionally, because DNA is allowed to wrap around itself, the enhancer region is actually pretty close to the transcription start site when DNA wraps around. Somewhere in chapter 29b, I don't know where, They describe they give you the name of the protein that causes DNA to bend. It is called DNA bending protein. Okay? Whenever you see that phrase, that is just a eukaryotic thing. How do I know it's just a eukaryotic thing? Well, eukaryotic DNA is the only one that can bend around like this. So eukaryotic organisms are the only organisms that have these DNA bending proteins. And they greatly aid in an enhancer's ability to interact with the basal transcription apparatus. All right, so back to DNA elements. Each one of these terms has a slight definition, and some of them are umbrella terms. I'll go over them, I believe, in the next slide. But for now, we know promoters. Promoters are pretty easy. Those are the regions right next to the transcription start site. Enhancers are further away. Response elements are DNA sequences where when a particular transcription factor binds to it, it is in response to some stimuli. We'll go over some examples in a second. Silencer elements are DNA sequences that when transcription factors bind to here, they actually inhibit transcription. And initiators are the opposite, which is the more common one. Initiators are sequences that when transcription factors bind to it, they initiate transcription. Again, you have general transcription factors that prefer the core promoter region. You have specific transcription factors, not listed here, but a separate category called specific transcription factors that go to the regulatory promoter region and to enhancer sites. Okay? So make sure you keep those two things in mind. It is not listed here, but other than general transcription factors, there are specific transcription factors. Specific transcription factors prefer the regulatory promoter region and enhancer regions. And then the last thing they want to mention on this slide is that there's a category of enzymes that exist to modify chromatin. The one example they give here is histone acetyltransferases. These enzymes attach an acetyl group to your histone, making it easier to transcribe genes in that region. Okay. More details about enhancers. Enhancers are far away. That one's easy. Transcription factors can bind to them. Also easy. Now, interestingly, with enhancers, is that they can both be upstream or downstream of the gene they regulate. That is in contrast with promoters. Promoters have to be upstream of the gene that they regulate. Enhancers can be upstream or downstream. Other interesting things. They are bidirectional and promiscuous. These two terms kind of mean very similar things, but I'm going to try to simplify it. Basically, an enhancer is so far away that because it's both upstream and downstream of a particular gene, it can interact with many different promoters simultaneously. Enhancers are able to work with many different promoters, whether in front of the enhancer or behind the enhancer. It can work with several of them. That's kind of what they were trying to say when they mean promiscuous there. And like I said earlier, specific transcription factors bind to enhancer sequences. That is stated here. Silencer sequences I already talked about. This is where repressor transcription factors bind to. Transcription factors, by definition, are DNA binding proteins. They can either increase or decrease transcription. Kind of a common mistake is people think that all transcription factors increase transcription. No, the definition of transcription factor is it's a DNA binding protein that regulates transcription in some way. Some of them are silencers. Here's response elements. Response elements, like I said, is a particular sequence that when a transcription factor binds to, it's in response to a particular stimuli. One example is the heat shock element. It's a particular sequence that heat shock proteins bind to, heat shock transcription factors bind to, in response to massive changes in temperature. Other ones, you have glucocorticoid response element, and I guess that means metallic response element. Basically, any of these elements, again, transcription factors will only bind to these things in response to a particular stimuli. Let's see. Oh, and this is a pretty interesting bullet point here. These response elements are constitutively expressed. They are found in many, many genes across the genome. And the reason why is usually when a cell is exposed to a particular stress, there is a massive response, not just one or two genes being turned on or turned off, but hundreds of genes being turned on or turned off to try to get the cell to survive whatever stimuli it's experiencing. So that's why you see here that response elements are necessary for the maintenance of basic cellular function, because they're trying to survive a particular stimuli. All right, let's see. This is post-transcriptional modifications. Okay. Way back in 10b, I believe, or 10a, we learned that eukaryotic mRNA is made initially as something called pre-mRNA, right? That pre-mRNA is indicated right here. Eukaryotic transcription makes pre-mRNA. That contains both exons and introns in them, right? Which one needs to be thrown out? The introns. The introns are your intervening sequences. They need to be thrown out in order to splice together the exons, and you can transcribe, and you have your finished transcript product. That whole process is called splicing, where you get rid of the introns and put together the exons. There's two other modifications, now excellently illustrated here. Two other modifications you see in eukaryotic mRNA, and that is the attachment of something called a poly-A tail, which is for stability, and the attachment of something called a 5' guanine cap, which is for the ribosome to have an easier time of finding where to bind on the mRNA. It's like a signal for ribosomes to start on that end of the mRNA before they start translating it. So these are the two modifications we see in eukaryotic transcription. This comes with after knowing that, you kind of have to think about some things a little bit deeper. There's some questions you get on the exam that's like, is it possible to clone a prokaryotic gene into a eukaryote? If you straight up just take out a prokaryotic mRNA and throw it into a eukaryote, will eukaryotic ribosomes be able to transcribe it? The answer is no, not straight up. If you take a prokaryotic mRNA and you throw it in a eukaryote, the eukaryote cannot translate it. And why? Well, that's because prokaryotic mRNA does not have a poly-A tail and does not have a 5' G cap. The eukaryotic ribosomes will not know how to translate prokaryotic mRNA because it's lacking these things. All right? Some other things to think about. And I know this is talked about more in chapter 30, so forgive me if I'm jumping ahead a bit. In prokaryotic mRNA, there's a particular named sequence on the 5' end that ribosomes like to bind to. Well, anyone know what the name of that sequence is that ribosomes like to bind to in prokaryotic mRNA? Shine-Delgarno sequence. You go review your, I think, chapter 29a somewhere they will mention that, or chapter 30 they will mention that. The reason why I'm bringing that up is that's a prokaryote thing. Prokaryotic mRNA has Shine-Delgarno sequences for prokaryotic ribosomes. That's another problem. If we take prokaryotic mRNA and we throw it in eukaryotes, can a eukaryote translate it? No, because a prokaryotic mRNA has a Shine-Delgarno sequence. The eukaryote doesn't understand what that is and can't translate it. So there's a lot of challenges to biomedical research and science when we try to get one organism to translate or transcribe another organism's stuff. So keep these details in mind, because those are more the styles of questions that are asked about on the exam, not just straight up memorization. You've seen these already in exam 1 and 2, that he sometimes wants you to think a little bit deeper about some of these concepts. I will review them some more when we get to chapter 30 and during the exam review, so stay tuned. All right, we already talked about general transcription factors a million times, so I'm going to skip that. We already talked about proximal and distal elements. Proximal elements are your promoters, distal elements are your enhancers. We talked about, well, activators are, I don't really like this term, because activators make it seem like there's a specific category of proteins called activators. No, activators is just a transcription factor that promotes transcription. That's it. You see where it has here, where it has two domains, one that binds DNA and one that activates transcription. In reality, every transcription factor has at least two domains. One that binds DNA and one that does something to transcription. Oh yeah, I guess if you don't know what a domain is, let me explain that. A domain is a particular spot on a protein that has a function. Well, you learned way back when we were talking about enzymes that enzymes have active sites and allosteric sites, right? Those are individually domains. When we talk about transcription factors, every transcription factor has a domain where the transcription factor binds to DNA. Again, what that means is that the transcription factor has a particular spot on it where it will bind to DNA, and it has another spot on it where it will regulate transcription. That's another domain. So domains are areas on proteins that have functions, are spots on proteins that have functions. Blah, 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 blah. Here's a visualization of these domains of a transcription factor. The ones on the bottom here bind to DNA, and the areas on the transcription factor up here do something to transcription. That's all they're illustrating here. Oh my God, they repeat things often so many times. These talks about silencer stuff. I'm going to skip that. We already talked about it. Some activators and repressors change chromatin structure. I already talked about that. We know histone acetylases, histone methyltransferases. We already talked about that. The loops in DNA are made by DNA bending proteins. If you want an illustration of what that looks like, here's that right here. DNA bending protein, bending DNA so that enhancers can interact with the transcription start site. Oh boy, there's plenty of information on here. Feel free to read it if you want. It is likely not going to be asked about on the exam. Skip, skip, skip. Let's see. Oh, homeobox proteins. Okay, let me look for a good illustration for this. Let's see. No, not that one. Where's a good explanation of that? All right. Well, forgive me. They don't give you step one, so I'll have to try to talk about it. Okay. I think in chapter 10b or 11, they talked about embryonic development and how chromatin is linked to embryonic development, right? This section of the notes kind of goes over that in more detail. So here's how it works. There's a set of genes in the body called Hox genes or homeobox genes. Okay, it kind of means the same thing. These genes are the ones that are the first genes activated during development. Way back when you have your zygote and your blastula and gastrula phases, that stage of development, your Hox genes are activated. What these genes are responsible for are creating the sections of the body, creating an orientation of which end of the body is going to be the head and which end is the tail for organisms. They're very basic genes that don't really focus on anything really specific, just basic genes that are necessary for the embryo to initially develop, okay? So only a certain set of transcription factors regulate these genes. These are embryonic transcription factors, which was very lightly touched upon in 10b or 10a when he was talking about chromatin. So homeobox genes or homeoproteins or Hox genes, when you have the umbrella terms there, are all talking about the transcription factors and the genes that drive embryonic development. Once an organism is fully developed and born and living their life, none of these genes are turned on. They're all off, right? Otherwise, you have weird situations in the lab when we mess with Hox genes. In an organism, sometimes they grow an extra limb. Sometimes we usually do this in flies. That's one of the reasons why they're called Hox genes. We mess around with fruit flies and we see that they grow extra wings or more eyes or less eyes. And sorry if this is disturbing, but this is what biomedical research sometimes is. But those are what Hox genes are. They give you they help the body make the kind of frame. They make the frame of the body. And so once an organism has been fully developed and is born, they're all turned off. So this act of switching from homeobox genes to other genes involves the activity of specific transcription factors. So I'm going to explain that. All right. In development, how does a stem cell know to become a liver cell? How does it know to become a lens cell? How does it know to become a brain cell? Usually this involves a switch from homeobox gene activation to specific gene activation. And what controls this switch is the turning off of Hox genes and the turning on of specific genes by specific transcription factors. This slide illustrates that from the same stem cell, you can form a liver cell or a lens cell based on what transcription factor binds to a particular site in the genome. You don't have to memorize the site, and you don't even have to memorize the transcription factor. Just understand that in development, there's a stage at which Hox genes are turned off, and we are starting to turn on specific genes by using specific transcription factors. So here's a lens transcription factor, not lens transcription factor, liver transcription factor that's going to start transcribing liver genes so that these stem cells become liver cells. Over here in the lens, you have transcription factor specific for lens genes, so that stem cell becomes a lens cell. All right. The one example they're trying to state here is that albumin is a liver gene, and so notice in the liver cell albumin is made. And in the lens, crystalline is a lens gene, and therefore notice that crystalline is being made only in the lens, but not in the liver. So they're showing this specificity is really important in the later stages of development when you get specialized tissues. All right, let's see. Oh, I guess they go over the details here. Feel free to look at it if you want. Don't worry about Cre-based cloning, which is what this is. Oh boy, blah, 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 blah. Okay. All right, coordinated gene expression. So, in eukaryotes, another kind of separate thing that eukaryotes deal with that prokaryotes don't have to deal with is that in eukaryotes, the genes necessary for a particular metabolic pathway might be on completely different chromosomes. As an example, the genes necessary to make the proteins for glycolysis are on wildly different areas in the genome. So how are they transcribed and translated simultaneously so that you have those proteins ready to break down glucose? The answer is all those proteins and genes probably respond to the same transcription factor. So coordinated expression of genes, meaning if you need a set of genes to be expressed simultaneously, likely need the same transcription factor to express them. We learned about response elements. That's more of a bigger version of this. Response elements are those response transcription factors that will turn on a set of genes to give the cell a chance to survive a particular environmental stimuli. But on the smaller scale, if you need like a set of metabolic proteins, just use one transcription factor and it'll activate the transcription of those genes. So that's a eukaryotic thing, is a coordination of transcription of multiple genes in completely different parts of the genome by using the same transcription factor. Let's see, we talked about loops in chromatin. I don't think you need to know more than that. Yeah, you don't really need to know more than that. This we talked about already before, the BAF blocker. This was earlier in the notes. This is just an example of what happens when chromatin, when you inhibit the ability of some enzymes to move chromatin around, you affect gene transcription. You can review the SI session when I covered that earlier. Okay, now we get into the actual enzymes. So all that stuff, which you see is a majority of chapter 29b, was how transcription is regulated in eukaryotes. That is the most important thing when we talk about eukaryotic transcription, is how it's regulated. The rest of it is talking about the actual enzyme that carries it out. Now we know in eukaryotes, there are 3 RNA polymerases, RNA polymerase 1, 2, and 3, and each one takes care of the transcription of one type of RNA. The one we'll talk about mostly is RNA polymerase 2, because this is the one that makes mRNA in eukaryotes. All right, so what do you need to know about it? Well, on RNA polymerase 2, it is composed of 2 subunits. Well, more than 2 subunits, but 2 important subunits. RPB1 and RPB2. You might find when you read the notes here that these are similar to beta prime and the beta subunit on prokaryotic RNA polymerase. If you don't remember what that is, you can review your prokaryotic transcription notes. And you will find that in prokaryotic transcription, those subunits were responsible for binding DNA and RNA nucleotides. So guess what? In eukaryotes, these 2 subunits are each responsible for binding DNA and RNA nucleotides. One interesting fact here, and very important for the regulation of eukaryotic transcription, is this thing. In eukaryotes, that subunit there, RPB1, has a C-terminal domain that can be phosphorylated or dephosphorylated. That one domain is kind of a switch for RNA polymerase II. We'll see in a second because I always get mixed up whether phosphorylated or unphosphorylated turns it on. But that particular site, when you affect it, you either are turning on RNA polymerase II or turning it off. So let me see, because I always get it backwards. Yeah, here we go. Only RNA polymerase II, whose C-terminal domain is not phosphorylated, can initiate transcription. So that one spot, that one domain, that one spot on RNA polymerase II, if it is phosphorylated, cannot perform transcription. Okay, so it is an on-off switch on that enzyme. So make sure you know that. That is often asked about on exam 3 or the final exam, if this is going to be on the final, is that domain regulates RNA polymerase II, either turning it on or turning it off. And you read what it says in red there. If it is not phosphorylated, it is turned on. More pictures, oh boy, blah, 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 blah. Again, a lot of these will be skipped, I guarantee it, when it gets to this section of the notes. All right, structural motifs, okay. This used to be talked about in detail 3-4 years ago. He's been talking about it less and less because of a mechanism called CRISPR. So forgive me if I go over this and he decides to not talk about this. All right, so I'll give you the simplicity of this. So, I talked about how transcription factors have domains in them, right? One domain that transcription factors have is something called a DNA binding domain. It is a particular pattern on the transcription factor. When you go look at the protein pattern in it, you go look up in a textbook and you see it'll say this is a DNA binding domain. So that transcription factor, just by having that domain, will be able to bind to DNA. A little bit more specifically, there's a variety of DNA binding domains that a transcription factor can have. Those are called structural motifs. Those are listed here in these colors here. There's more than what's listed here, but these are a majority of them. There's a particular type of DNA binding domain called a helix-turn helix. There's another one called a zinc finger, and another one called a leucine zipper. These are particular categories of DNA binding domains that transcription factors can have. And depending on which one they have, it'll regulate which areas in the genome it likes to bind to. Okay? You don't have to memorize which one allows a transcription factor to go to here versus here. Just know that these are varieties of structural motifs, varieties of DNA binding domains that a transcription factor can have. Let's see. So they go over them in detail. Feel free to read them if you want, but for this class you don't need to answer anything specific about structural motifs. Just recognize by name the structural motifs. Zinc finger is usually the one talked about the most. It's the most common type of structural motif that transcription factors have, so that's why you'll see probably a little bit more detail on the zinc finger motif thing. But I'm just going to move along. And this is visually what the two forms that a zinc finger can have. So a particular transcription factor, if it has a zinc finger motif, somewhere on it it'll form this shape like so. And that shape actually is very favorable in terms of DNA binding. This is the other shape it could possibly form. More zinc finger stuff, more zinc finger stuff. Okay, now the important part. So, the reason why he doesn't talk about structural motifs a lot anymore is because of this technology called CRISPR. Now, naturally speaking, natural transcription factors will do their job via those structural motifs that we talked about. They have either zinc fingers, leucine zippers, whatever, and they will find a place to bind to and transcribe it. Lab-wise, if we want to transcribe genes artificially, we used to have to design proteins or modify proteins that had zinc finger motifs on them. And that became a bit of a difficulty because if we make a protein that's a zinc finger protein, it might not go to the spot on the genome we want it to go to. Only recently, within the last few years, there's a technology called CRISPR that has been developed. Oversimplification, But what CRISPR is a way to specifically edit the genome. It's to specifically edit sites on the genome. And here's how it works. Okay, so there's a lot of pictures on here, and I'll point out some things that you need to know at the end, but here's how it works. Here's a complex here, a set of protein holding something. It is holding this purple strand of DNA. That is called a tracer sequence. They might use a slightly different term on this slide, but I learned it as a tracer sequence. This tracer sequence is what the protein is holding on to, and what it's going to use it for is it's going to find a spot on the DNA where this tracer sequence matches to. Okay, so that's what this is illustrating. It's holding onto this purple strand of DNA, this tracer sequence, and is searching the entire genome of whatever you're studying, and it's going to find a place to match to. When it finds that spot, it's going to cut that region of DNA. It's going to open it up. Whenever you open up a spot in DNA, you can insert whatever you want in that spot. So this technology allows you to do specific gene editing, to go exactly where you want to in the genome, to cut it open, and then the easy part is inserting whatever you want in there. The difficult part historically has been getting components to specifically open up a particular spot in DNA. You might remember back in chapter 11 that we learned about something called restriction enzymes, right? Restriction endonucleases. Those were the also old-fashioned way that we edited DNA, right? If we had restriction enzyme EcoR1, it would cut open places in DNA that have an EcoR1 site. But we were limited because if we use that restriction enzyme, it's only going to go to places in the genome with an EcoR1 site. What happens if we want to edit a gene that doesn't have an EcoR1 site? We'd be lost. We wouldn't be able to edit that gene. The reason why CRISPR is so amazing, mechanically speaking, is because we can design this tracer sequence from scratch. We can make that tracer sequence from scratch. So if we know a particular gene we want to edit, we can make the tracer sequence that matches to that area in the genome, throw it together with the CRISPR system, and it's going to go exactly to that spot and cut it open. And we can insert whatever DNA we want in there. Currently, I don't know if it's been FDA approved yet or something, there's a set of therapies called CRISPR therapies that are being developed. Basically, right now, at the very beginning stages of this technology, they're trying to edit genes that are the cause of debilitating genetic diseases, okay? So by using CRISPR, if there's a particular mutation in a gene that would normally lead to some horrible disease later in life, we would use this CRISPR editing function to go and modify that gene to remove that mutation so that very likely that patient will not develop a debilitating genetic disease. So that's the mechanical side of it. What you would be asked about, of course, is to be able to label the certain things you see on this picture, to understand what it is used for, and that is gene editing, specific gene editing, and then you'll be good in terms of understanding what the CRISPR-Cas9 system is. So make sure, you know, if you're given a picture like this and it's pointing to various things, you can label whatever's labeled on here. You'd be able to identify it on the exam. Let's see, what else did I want to mention here? Sometimes they'll go over some interesting philosophical stuff about CRISPR-Cas9 and gene editing. You won't be asked about that on the exam, but if you are a curious scientific person, it actually is a very important thing to think about, because if all of a sudden humanity has the ability to edit genes in whatever way they want, is that a good or is that a bad thing? So keep that in mind. A lot of people immediately think about the good stuff, but there's also a lot of bad stuff that can happen, right? Hey, there's plenty of sci-fi movies that already exist that say what could possibly happen, but in reality, there's a lot of potentially bad things that could happen with this. All right. And you know, as an informed, scientifically thinking voter, keep that stuff in mind in case that ends up being some policy issue in the future. All right. We already talked about post-transcriptional modifications in eukaryotes, so not going to bother. Let's see. Already talked about introns and exons. Man, so much to skip. GCAP, skip, skip, skip. Let's see. Poly A tail, skip. Terminator signals. Nope. Okay. Oh, there used to be. Oh, I guess he doesn't want you guys to know that anymore. Awesome. All right. Okay. So I guess that's actually it for 29B is those basic concepts. And that, again, will potentially be on exam 3 or not. I will let you know when it comes time for the exam review on Wednesday evening. At minimum, this will definitely be on the final exam. All right? So we'll end a little bit early today. I will start reviewing for exam 3 in the final next SI session. So I'll see you then. Yes?